Today, I have the pleasure to introduce Dr. Celina Juliano, who has an indirect connection to Carnegie. Dr. Celina Juliano is an assistant professor in the Department of Molecular and Cellular Biology at the UC Davis. Her lab studies development and regeneration in the aquatic animal Hydra, which undergoes continual self-renewal, lacks senescence, and has incredible regenerative capacities. Dr. Celina earned her PhD at Brown University working with sea urchins. During her graduate work is also when she learned what a cool organism Hydra is during a talk given by Rob Steele. She contacted him and started doing research with Hydra while still in her graduate student lab. She used that data to get a fellowship and continued her research in the Hydra at Yale with Hai-Fen Lin, who has a Carnegie connection and just recently gave a public lecture. So I'm very excited to hear about Dr. Juliano's research, and welcome.
Okay, can you hear me? Hold on, let me arrange myself here. Okay. Yeah, so I have an hour to talk and I don't really want to talk to myself for an hour. So I put some breaks in so that I can answer some questions along the way. So thank you very much to Natalie and the rest of the postdocs and the students for this invitation and for organizing this symposium. I really do wish I could be at Carnegie in person. I've never been and I have heard great things about it, but I'm grateful that we can at least do the symposium online, and I've been really enjoying it so far. So I wanted to start my talk today by telling you why Hydra is so great. Like, why is this a great model for studying stem cells and regeneration? Oh, there we go. Okay. So I always start with this video because it really shows the power of regeneration in Hydra. So what we're looking at here is a small piece of tissue taken from the body column of the Hydra. This is about 100 micron in diameter tissue. And that small piece of tissue can regenerate into a completely functional organism within 2 days. So we would, really like to understand how that happens. So Hydra is a cnidarian related to animals such as sea anemones, corals, jellyfish. These cnidarians are mostly in the ocean. There's saltwater organisms, but Hydra is one of the few that made it into the freshwater. That makes it a little easier to take care of in the lab. So cnidarians are have radial symmetry, and so they're organized around a single oral-aboral axis. At the oral end, we collectively refer to these structures as the head in Hydra. This includes tentacles for catching food, and this hypostome structure here, which includes the mouth for eating food. It's actually the only opening in the animal, so this is where it excretes waste. At the aboral end is the foot structure, which includes this sticky basal disc. So the Hydra, when it's out in the lakes and streams, it sticks to leaves and rocks. In the lab, it sticks to petri dishes. And then just above the basal disc is this tissue we call the peduncle. So all the rest of the Hydra is this hollow body column that connects the head to the basal disc. And it's the body column is where the food inside is where the food is digested, and the body column tissue is the regenerative tissue, and this is where all the stem cells reside in the organism. So you may notice this cute little Hydra coming out the side here. This is called a bud. So the Hydra can reproduce asexually by making these little genetically identical clones coming out from the side of the body. This happens always in this area called the budding zone. And so if you have one Hydra that you like, you can make an indefinite number of copies of that Hydra. And so that's really convenient for the lab. That's largely how we propagate the animals. We do this with asexual reproduction. Luckily, the animals can also undergo sexual reproduction. It's a little bit more finicky, but it does happen. And in this case, the females make an egg from the side of the body, and the males make these testes structures, and then the sperm can come over, swim over here, and fertilize the egg. And so these eggs are really easy for us to access, and we can inject these eggs with plasmid DNA. And actually, it turns out the Hydra genome just takes up DNA quite easily, and so we can get transgenic animals actually pretty readily just by injecting these early embryos. And this is an example of a transgenic Hydra. So in this case, we have GFP expression in the outer layer of cells and RFP expression in the inner layer of cells. And I should point out that although fluorescent proteins were originally discovered in cnidarians, luckily for us, Hydra doesn't have any natural fluorescent proteins. So these are ones that we have added to the animal. I like this particular transgenic line as a way of showing you the basic structure of Hydra, which is two epithelial monolayers separated by an ECM. So we call this outer layer the ectodermal layer, and the inner layer, the endodermal lineage. Okay, so I'm going to switch now to drawings of the Hydra. So that can really give you a better idea of the cell types and the structure. So these are the two lineages that I just spoke about. So the outer layer is the ectodermal lineage, and the inner layer is the endodermal lineage. So I've colored the ectodermal cells blue and the endodermal cells green. All of the cells in the body column, which I've colored in more lightly, are mitotically active. And so these cell divisions push the ectodermal endodermal cells towards the extremities. And then when these cells get to either the oral end or the aboral end, they terminally differentiate to create the tentacles and the hypostome or the basal disc. And then the older cells are shed. And so in this way, the animals in this continual state of development, with the older cells being lost and newer cells always being made to replace the older cells. So these are two lineages in the animal, and then the third lineage is the interstitial lineage. And these cells reside within the interstices of the epithelial cells. So if you take a cross-section of the body column, for example, you'll find all of these little cells interspersed within the epithelial cells. So this lineage is supported by this undifferentiated multipotent stem cell called the interstitial stem cell, which resides in this ectodermal layer. There's a few different cell types that are part of this lineage. So one is the nematocytes, which are the stinging cells of Hydra, but also of all cnidarians. And these are embedded into the ectodermal layer, largely in the tentacles, but some are found in the body column. And so these help the animal defend itself and catch food. There's also the neurons, which form nerve nets that go throughout the animal. There's a nerve net embedded in the ectodermal layer and a nerve net embedded in the endodermal layer. And then the other kind of cell type is a gland cell. This is strictly in the endoderm, and these cells will secrete digestive enzymes into the body column to help digest food. So the interstitial cells are closely associated with these mitotic epithelial cells. And because of that, they are being pushed along as well and are often lost as well. And so actually every 20 days, all of the cells are basically replaced. And so this is, again, this constant state of development and renewal, just in a homeostatic animal, not even one that's regenerating. So because the cells are constantly being moved around, they need to know where they are at all times. And this can occur because there's positional information emanating from the oral and aboral ends through signaling, basically. So a lot more is known about the oral organizer than the aboral organizer. So I'll just give you one slide to discuss the oral organizer. So the oral organizer was initially discovered by Ethel Brown in 1909. And what she found is that if you dissect the hypostome, which is the tissue just at the oral end here, and you transplant it to the body column of a different organism, you actually get the growth of an entirely new axis. And in this example, you see this is a non-transgenic hypostome, which has been transplanted onto a transgenic GFP-expressing donor, or acceptor, sorry. And that non-transgenic tissue organized the transgenic tissue into a new axis. So that's why you call this an organizer, because it can organize the fate of other tissues. So this was discovered before we knew anything about Wnt signaling, but developmental biologists probably wouldn't be surprised to hear that it's the hypostome is a Wnt signaling center, and it's really Wnt signaling that's responsible for the oral organizing capability of the hypostome. So this was first found by Bert Hohmeyer and colleagues in 2000, that there are Wnt's expressed in the hypostome, and has since been demonstrated by several methods that the Wnt pathway is both necessary and sufficient for oral fate, for creating heads. And so this is one such experiment where a stabilized beta-catenin is being ectopically expressed throughout the animal. The output of Wnt signaling is essentially to stabilize the transcription factor beta-catenin to go into the nucleus to act with TCF to activate genes. So if you make a stabilized beta-catenin that cannot be degraded, what you get is a Hydra that's full of lots of extra heads. And so this is one of the ways to demonstrate that Wnt signaling is sufficient for head fate. We know that you can transplant the aboral end and get a new foot growing at the side of an animal, but that signaling center is actually not at all defined in Hydra. Okay, so that's sort of the end of the part where I just described stuff about Hydra. And so I wanted to maybe, if there are any questions at this point, just about Hydra generally, I could answer them, and then I'll go on to talk about the research in our lab.
So I see one question.
Okay.
John Sidman asked, what is the timeline for transformation?
Okay, that's a great question. So the way that transformation works is that we inject plasmid into the egg. It takes about 2 weeks for the embryo to develop, and then it goes into, then it hatches out, essentially. So it goes into this cuticle stage, and after 2 weeks from point of injection, it hatches out. Once it hatches out, we have these little tiny Hydra. They look just like a big Hydra, but they're smaller. And at that point, we'll see some random integration of plasmid in between 30 to 50% of the animals we inject. Could be a very small patch, could be a bigger patch. Now, because they undergo asexual development, we would just let that first animal asexually develop so we can propagate the transgenic tissue that way. And then we try to select buds that are more and more transgenic. And it will just be for one of the 3 lineages, usually not multiple lineages. And so then, depending on how much transgenic tissue we had in the first place, usually within a few months, we can get, a line of actually usable animals. Okay, is there any?
We have some more questions.
Yes.
Amy Herbert asked, are the interstitial cells post-mitotic, and what are their lifespans?
Okay, so it depends on the cell type. So for the neurons, once they are differentiated, they are post-mitotic, and they're all gone within about 20 days. So how they are removed is a little bit of a mystery, but there's definitely turnover. And I'm going to actually get to the nervous system a little bit later in the talk. For the gland cells are actually not post-mitotic, so they can renew themselves, and not very many have to be made from the stem cells themselves because they are continually mitotic. The nematocytes, they are post-mitotic. They're a very highly differentiated cell, and they're basically single-use cells. So once a nematocyte fires to catch a prey, for example, it has to be discarded and then remade.
The next question is from Yuko Ota. I thought there is a histocompatibility matching between different individuals. Can you graft head of any individual to others?
Yeah, so you can. As long as it's the same species of Hydra, you can do all kinds of cool grafting experiments. It doesn't have to be the same genetic background. As long as it's the same species, you can mix and match them any way you want. So there's lots of cool things you can do because of that.
Yixin Deng asked a question. I remember reading that aggregation of dissociated single cells leads to regeneration too. Is this true? If so, where is the position signal?
Yes, great question. Yes, it's true. I'm not going to talk about it at all in this talk, but there's people starting to look at this more and more, because it's a really cool thing for cell sorting and how do you set up signaling centers from scratch. So essentially you can dissociate Hydra into single cells, mix it all up. Usually you do it with a large group, like, several, at least 10, if not 100 Hydra, you dissociate them all. And then you can make little balls of cells. They're all mixed up. Within a few hours, all of the ectodermal cells will be on the outside and all the endodermal cells will be on the inside. So they sort out, and then new signaling centers will arise. Little Wnt signaling centers will come up around the ball, and then new heads will pop out. So how do the Wnt signaling centers get set up? We don't know. I think this is a really great question. Somebody should be investigating that.
Last question. I hope I pronounced the name right. Zirong Wang. Does constant cell renewal mean Hydra is essentially immortal?
Yeah, so the short answer is yes, although I hesitate to use the word immortal because we can't really test that it is immortal. But lots of longevities, well, not lots, some really impressive longevity studies have been done. The longest one was 8 years of basically just tracking the same Hydra, individual Hydra over time, counting buds and removing buds, but keeping the original animal. And over 8 years, there was not much mortality at all. And they projected that the lifespan could be anywhere from 100s to a few thousand years. So it's difficult. It's a difficult thing to really study, the aging of an animal that doesn't appear to age. But the short answer is, yeah, it seems to lack senescence and perhaps be quote unquote immortal.
We have one more question, but I will ask it in the next question round.
Okay, sounds good. Thank you. Thank you for all the great questions. Okay, so now I'm going to go on and talk a little bit about, or a lot, I guess, about the work going on in our lab. And so our sort of big picture idea is that, so we have this animal that is even in its homeostatic state, is in a continual state of development, and also it regenerates really well. So it's a great developmental biology model for just how do cells differentiate, how stem cells work, and also a great model for regeneration. So we would like to understand the signaling pathways that are active, the gene regulatory networks that are active in the homeostatic animal. How do signaling pathways activate the GRNs, and how do those lead to cell differentiation in the homeostatic animal? And one of the reasons that we want to understand that is because then we want to also understand how does the conserved injury signal actually trigger these developmental pathways to turn on, at the right place at the right time during regeneration. I mean, in that question about how does a signaling center even set up when you completely destroy, everything and put it back together, could all kind of be encompassed also in those questions. So for the first part of the talk, I'm going to focus on just an uninjured homeostatic animal. And then at the end, I'm going to show you some actually new unpublished data we have addressing this question of how does the conserved injury response actually trigger, in this case, the Wnt signaling pathway. Okay, so one of the first big projects that my lab embarked on when we started was a single cell RNA sequencing project. So of course, everybody is doing this now, and it's the hot technology. I was really excited about single cell RNA sequencing when I first heard about it, because my sort of initial thought was that, okay, Hydra is actually a pretty simple organism. It doesn't have that many cell types. wouldn't it be great if we could just have a complete gene expression map, and then we could stop having to do like a billion in situs all of the time? We just go to the computer and ask, where is that gene expressed? And so we actually have that now, and it's really great. And also, you know, by morphology, we could see how many cell types it has, but how many molecular cell types are there was another question. And then what really came out of it that was really quite exciting was the fact that since Hydra is in this continual state of development, that just by doing single cell RNA sequencing on a single time point, the adult, we could actually build lots of differentiation trajectories. And so I have like this really kind of toy example of differentiation trajectories for people who don't think about this too much. But this is, in my mind, why single cell RNA sequencing has really been a great thing for developmental biologists. So if you imagine you take a Hydra, you dissociate that into single cells, you get a whole bunch of different cells in different states. You have stem cells and you have differentiated cells, but you have these cells that are in the process of differentiating, and you've captured them in that sort of intermediate state. And so, you know, really smart people have developed all of these great methods for computationally ordering these cells based on the progressive changes in their transcriptomes into these differentiation trajectories. So this is a little toy example where you have a stem cell and you can see it differentiating into 3 different cell types. And what's one of the things that's really great about this is you can focus in on these branch points, and you can ask what transcription factors are expressed at these specific branch points. And you could hypothesize now that these are transcription factors that are really important for specifying particular fates. Okay, so this study is, we published it in July of last year. Just wanted to, before I talk a little bit more about it, give credit to all the people who worked really hard on this study. This was led by a project scientist in my lab, Stefan Siebert, who recently took a great paying job in the Bay Area at a startup company. And we also had tremendous input and help from Jeff Farrell, who was a postdoc at the time at Harvard, but has just started his lab at the NIH, and he's a fantastic collaborator. We have 2 graduate students working on this project, Jack Hazet and Abby Primack, and I'll sort of show you along the way where they came in. And then our former lab manager, Yash, who's now a graduate student at UCSF, she did a huge number of in situs to help validate some of these data. Okay, so this is the typical single cell t-SNE plot that people will show. So I'm not going to make you look at all the details of this, but I just want to kind of give you an overview before I start getting into some of the data. So we sequenced about 25,000 cells using DropSeq. This is a microfluidics-based single cell sequencing approach. Our statistics are that we got about 2,000 genes per cell and about 5,000 transcripts per cell. So of course we don't capture the entire transcriptome of every cell, but when you sequence enough cells, then you get a lot of information, and you can kind of infer the transcriptomes of each cell type. So we cluster the cells together using principal components analysis, as everybody does. We use the t-SNE dimensionality reduction. So this is all done with the Seurat-R package. And then we were able to annotate all of the cell types. So that's what you're looking at here. It's just all of our clusters and all the annotations that we could add here using lots of in situ patterns. So we have the luxury of looking back through the literature. There's lots of in situ patterns available. We also had to do some of our own validation. Okay, but there's not a lot you can learn just by looking at a t-SNE of an entire animal. So we're going to break it down a little bit. So the first thing is that we extracted each of the 3 lineages from the data set to analyze that individually. So this is meant to show you that for the 3 lineages, endoderm, ectoderm, interstitial, we have very good representation of all 3 lineages. I'm not going to discuss today the endoderm and ectoderm lineages. They're very interesting, but for the sake of time, I won't discuss every single lineage. I'll focus on the interstitial lineage, which is maybe the most interesting, but this is published, so please feel free to look that up if you're interested. So we extracted all of the interstitial cells. This is just to remind you of the interstitial lineage itself. So as I had mentioned, it has a multipotent stem cell. This is kind of what we knew already about the interstitial lineage from the literature. So interstitial stem cell, it can give rise to the germline, which I didn't really talk about, but it can. There are male and female germline stem cells in the animal, but if those are depleted, then these can recreate those germline stem cells. It can give rise to 4 different kinds of nematocytes. It goes through these incomplete cell divisions to get these nests of cells, which then burst apart and become nematocytes. It can become 3 different kinds of gland cells. And then for the nervous system, by morphology, there's 2 obvious types of what the Hydra biologists refer to as the sensory neuron and the ganglion neuron. But by molecular types, we already knew there was definitely going to be more than 2. Okay, so we extracted all of the cells from all the interstitial cells, and we reclustered those, subclustered those. And what you can see already in the t-SNE plot before we even start building differentiation trajectories is that you have a population of stem cells, and you can see neurogenesis happening. These are cells undergoing neurogenesis, and these are cells undergoing nematogenesis. We could sort of figure this out from particular genes that were expressed. We have the nematocytes here, lots of populations of different neurons, some gland cell populations, and then germline populations. Okay, so we worked with Jeff, as I mentioned, Jeff Farrell, who is the one who developed the URG software package to create differentiation trajectories. He developed his software package to work in zebrafish initially, during zebrafish embryogenesis, where you have time points that you collect during embryogenesis. So we had to sort of retool things to work in a system where you have one time point, but you have all the different states in that one time point. So the short story is that you define a root, and then which is the stem cells, and tips, which are the differentiated cells, and then the software package connects the cells with similar gene expression using simulated random walks. Okay, so this is the result of building the differentiation trajectory. So here again, every dot is a cell. They're different colors because they've come from particular clusters. The stem cells are at the top, and then there's differentiation going down towards the bottom. We call that pseudo time. It's time, basically a time that we can't put a number on, but we know it is differentiating from stem cells to the different kinds of differentiated cells in the interstitial lineage. So we can see our 4 types of nematocytes, several different neuron populations, also gland cells. The gland cells are a little bit complicated, and I'm not going to get into it today, but I'm always happy to answer questions about that. But gland cells are doing a lot of transdifferentiation, which complicates this analysis. Okay, so one thing that we saw that was unexpected in this differentiation trajectory was the possible existence of a progenitor cell which has the capability of giving rise to either neurons or gland cells. So this was not something that was known before in Hydra. And again, this is really just a hypothesis still. We haven't proven that this is an actual cell that exists, but now we have the tools to actually test that. Okay, so I could revise, possibly there is this progenitor cell which is going through this transient state of being competent to either make a neuron or a gland cell. The reason we really like this hypothesis is because, as I mentioned at the beginning of the talk, in the ectodermal layer we have the multipotent interstitial stem cells. In the endodermal layer we have neurons and we have gland cells, which need to be replaced quite often. And so we know that progenitor cells have to cross over the ECM in some cell invasion event in order to replace these cell types. And so we hypothesized that the cell that crosses over is something that is already partly differentiated and is now a neural gland progenitor and has lost the ability to make nematocytes. So that's something we're interested in testing in the lab. So this here shows the power of now trying to identify transcription factors that might be important in that neural gland progenitor. So here again is our trajectory. Now the cells are colored based on gene expression. So if they're gray, they're not expressing the genes that we're looking at. We're looking at 2 transcription factors, MYC3 and a MIB domain containing transcription factor. So the cells where we detected MYC3 are green. The cells where we detected MIB are this pink color. And then if they're black, we detected both of those transcription factors. So we did fluorescent in situ hybridization, and you can see these green cells here expressing MYC3, and then they're also expressing MIB. And so we found throughout all of our experiments that the MYC and MIB really are co-expressed genes, and we would hypothesize that these are the cells that have both neuron and gland potential, although we haven't tested this yet. Okay, so our hypothesis is that there's an interstitial stem cell in the ectoderm, which starts to differentiate, turns on MIB and also MYC3. If it stays in the ectoderm, it would differentiate into a neuron, but if it goes through a cell invasion event and goes into the endoderm, then it could supply the endoderm with gland cells and also with neurons. We do detect these MIB positive cells both in the ectodermal layer, shown here, and down here in the endodermal layer, suggesting that they do cross over. We have a new postdoc in the lab, Ben Cox, who's really gonna look carefully at this cell invasion event, try to capture it in the live animal and try to understand how that works. So stay tuned for that. Okay. So I showed you that we can pick out transcription factors expressed at interesting times, and we can start asking questions about them. But we also wanted to develop a way to more globally interrogate the data to try to find, out of all the 450 or so transcription factors in Hydra, which ones should we go after, which ones should we test. And so graduate student in my lab, Jack Kazette, made this strategy for trying to find the most important transcription factors that drive differentiation. And so he employed something called non-negative matrix factorization, which was not developed for single cell data, but people have been using for analyzing single cell data. And it works really well to identify groups of co-expressed genes in a completely unbiased manner. And so he identified 54 groups of co-expressed genes in Hydra, and that's all published in the paper. So here's some examples. So this is 3 of the 54. These are called metagenes. They could be anywhere from tens to hundreds of genes that are all expressed in the same time place. So here's one that's expressed in neurogenesis. Here's one that's expressed largely during gland differentiation, and then here's one that's specific to nematogenesis. So we have several, for example, that are specific to nematogenesis, and overall we have 54. So the next thing that Jack did was he looked into the regulatory regions of these co-expressed genes to look for enriched motifs. So that helps you try to figure out how these genes that are expressed at the same time and place are turned on. And we also have an attack-seq data set, which just helps you identify the open regions in the regulatory in the DNA to help you narrow down which motifs you actually look at. And then we tried to identify transcription factors that could bind those motifs that were expressed at the right place, time and place. So I'll give you one example. So in case you couldn't really follow what I was saying, I think an example helps. So we have a metagene or an NMF module of 73 genes that are expressed during nematogenesis. From the attack data, we extracted 73, it's just coincidence, it's the same number, reproducible peaks within 5 kb of the transcription start site for all of these genes that are expressed in this metagene. And then we looked for an enriched motif, and the top hit was a PAX motif. So the PAX motif is enriched in the regulatory regions of these 73 genes. And there is a PAX2A expressed during nematogenesis. And so we would hypothesize that PAX2A is required for turning on lots of genes during nematogenesis. What's nice is that actually Leslie Babonis and Mark Martindale's lab demonstrated already that PAX2A is important for nematocyte development in Nematostella, another cnidarian. And so this seems to be a conserved function for this gene in cnidarians. Okay, and so the bottom line is that we now have a huge table of transcription factors that could be important in each of the cell types. And so this gives us and the entire community kind of a starting point for identifying transcription factors and gene regulatory networks that drive the specification of each cell type in Hydra. So I wanted to take that as another stopping point before I'm going to talk about the nervous system of Hydra next. So if there are questions to this point, I could take them.
Hi, we have one question from the previous session.
Oh yeah, I'll answer that.
It's actually very relevant. Gonzalo Artigas asked, is it known whether adult interstitial cells are always dividing asymmetrically?
Oh, no, so the evidence right now in the literature would be that they could divide asymmetrically or symmetrically. And I think that that's pretty solid evidence. So that, yeah, they could do either. And they could make two stem cells, they could make a stem cell and a neural progenitor, or they can make a symmetric division that goes on to make two neurons, for example. But I think that with the tools we have now, we can start to interrogate that question like more carefully, because we can start to make transgenic lines that actually mark specific progenitors, which we couldn't do before. So I think there's lots of exploration still to be done on that kind of question.
Matt McCoy asks, maybe you will talk about this later, but are neurons the most diverse cell type in your single cell analysis? In other words, are there more subtypes of neurons and subtypes of other cells?
Yeah, that's a great question. So I am about to talk about the nervous system, but yes, it is true that those molecularly most diverse cell type is the neurons, and you're going to see momentarily what they look like. It's not as nearly as diverse as other organisms' neurons, I'd say, or non-cnidarians. Maybe bilaterians have more diverse neurons, but it is still the most diverse set of cells in the Hydra.
Nicole Lindsay Mosher asks, the metagene analysis is very interesting. Can you identify similar batches of genes activated during regeneration?
So we haven't used that analysis. I'm going to talk about regeneration at the end of the talk, and we haven't used that analysis on our regeneration data. We haven't, nor have we done single cell analysis on regeneration, but it's an interesting idea. I will pass it on to my student.
John Sidman is asking, if you dissociate the Hydra cells and have markers for cell type, do you know if cell types keep their identity when reforming?
Yeah, so we actually do know that for some of the cell types. So for endoderm and ectoderm, it's absolutely certain that they keep their fates, because we've done that with, we as a community have done that with transgenic lines. What's happening with the interstitial lineage is a little bit less clear. Possibility could be that all of the differentiated cell types die and are replaced by stem cells. The other possibility is that neurons, for example, could just like stay intact, reconnect up, and start making nerve nets. But that we have the tools to look at that, but it hasn't really been figured out yet.
Minje Hu is asking, are the transcription factors well defined in Hydra?
Okay, so what we have done, it's a well defined, I'm not sure how to say that. Maybe the answer is no. But what we have done is, what Jack did was he went through our entire transcriptome and pulled out anything that had a DNA binding domain. So clearly that could include things that are not really transcription factors, but that's like the best we have at the moment.
We have no other questions currently.
Perfect, okay. Thank you again for the great questions. Okay, so now I want to talk a little bit about the nervous system. So no one in my lab is a neurobiologist, but we're sort of dipping our toes into these waters. It's a little bit scary sometimes. It's a very huge insane field, but we are doing our best. So I'd like to start, just to impress you, with a video from Christophe Dupree when he was a graduate student with Raphael Yuste at Columbia. This is a transgenic hydra expressing the fluorescent calcium indicator GCAM6 in the interstitial lineage, which includes the neurons. And so you can see that you can image the whole hydra while it's behaving, and you can, watch the nervous system be active in this freely behaving animal. So in their paper in 2017 in Current Biology, they started defining some of the neural circuits and connecting those to behaviors. And so I think the dream of the neurobiologists that work on Hydra would be to know all the different neural circuits, connect them to behaviors, and kind of have an understanding of the whole nervous system. So I just want to tell you a little bit about the structure of the nervous system. So it is, as I mentioned, a diffuse nerve net. It runs throughout the whole animal. There is a higher density of neurons at the oral and aboral ends. In this video, you can really see the higher density at the aboral end here at the foot very clearly. It actually has two separate nerve nets. So one is embedded within the ectoderm and one is embedded within the endoderm. And to date, there's no evidence that they connect or talk to each other at all. The neurons are continually replaced without disruptions in behavior. So I find, as a developmental biologist, I find this interesting. And I think for neurobiologists, that's also interesting. Every 20 days, it's a new animal, and that includes the nervous system. And so it has to continually be removing and adding neurons into the system. And then, as I mentioned, in progress from labs such as Raphael Yuste and Jacob Robinson at Rice, they are working on identifying neural circuits and looking at their associated behaviors. We're helping out, and because we are identifying sort of all the different subtypes of neurons, and we are providing the tools to really control the nervous system in Hydra. And so I think what we're really excited about is that Hydra offers this opportunity to understand a highly regenerative nervous system from molecules to behavior. So we know, C. elegans, for example, is a fantastic model for studying the nervous system, but it's not regenerative the way that Hydra is. So this, I think, adds a new source of excitement for adding this new sort of simple system of studying the nervous system, but this one has the regenerative capability. Okay, so to start, we wanted to make a molecular and spatial map of the nervous system. So mostly what I'm telling you about today. So we took all the neurons out of our data set and reclustered, subclustered those. And so these are the neurons of Hydra. So we start to identify those. The ones in the center are the progenitors. So they are expressing that MIB and MIC3 that I spoke about. And then there's 12, we found 12 subtypes of differentiated neurons. If we are sequencing more to see if that refines even more, but at this point we have 12. So the first thing is that we wanted to see, are there distinct subtypes in the endodermal versus the ectodermal nerve net? So an interesting little tidbit is that previous to our work, actually, we knew that there were neurons in the endodermal layer, but no one had found any molecular markers that identified those neurons. They were a bit of a mystery. And so we were anxious to see if we could find any. So what we did is we took advantage of this kind of insane procedure that people developed in Hydra some time ago, where you can take the ectoderm and endoderm apart. So you put the Hydra in an acidic solution, and if you're really patient, you can roll the ectoderm down off of the endoderm. So this is the rolled up ectoderm, and this is the endoderm that came from inside the animal. And we confirmed that when you do this, the neurons stay attached. And so we just sequenced this half and we sequenced this half, and then we did differential gene expression analysis and also compared that to our neuron subtypes. And we found quite clearly that neurons are either in the endoderm or in the ectoderm. There's different distinct subtypes in each layer. We found only 3 subtypes that are in the endoderm, which made some sense too, because it's just a less dense network in the endoderm. We named these N1, N2, and N3, endoderm 1, endoderm 2, endoderm 3. And then we set out to validate that they really were in the endoderm. And what we discovered is that the reason why no one has ever found markers for the endodermal neurons is that in situs just don't work very well for endodermal neurons, and it was hard to actually validate these. And so we turned to making reporter constructs in transgenics. So I'll show you one example. So we found that this transcription factor, NDF1, is expressed very specifically in the N1 population. So we extracted the regulatory region, hooked it up to GFP, and made a transgenic line. And then we could see, we were very happy this day, that really these are endodermal neurons. So this is, the red is the ECM, the ectoderm is on this side, the endoderm is on this side, and you can see neurons strictly on the endodermal side. And we confirmed by transgenics the other two populations as well. So also, we have 9 ectodermal populations. Some are, so we have them also, I won't go into all the details, but we have them labeled by numbers and sometimes numbers and letters for reasons we could discuss if you're really interested in the nitty gritty. But we could use in situ hybridizations to test and validate all of these populations, some from the literature, some from ourselves. So this is a beautiful in situ from literature from Hansen et al. Of a neuropeptide that's expressed in the neurons just in the peduncle. And so this is this population here, which is called the EC5 population. Here's another in situ of neuropeptide that's expressed actually highly in the basal disc, also in the tentacles and head. And then it's harder to see, but throughout the body. And that's all of these cells here. And you can see that this population also splits into 3 separate populations, and we do have distinct markers that we've confirmed for each of these populations as well. Okay. And so I guess I would say that now we have this map of the Hydra nervous system, very specific. We have specific markers for every single one of these subtypes. We have the ability to express transgenes in every one of the subtypes. And so as we move forward with our neurobiology collaborators, we hope to manipulate the nervous system very precisely. So express things to remove particular subtypes, to activate specific subtypes, GChem expression in specific subtypes. So we hope to really dissect this nervous system. And then as developmental biologists, we're interested in how all of these neurons are actually made. And so I'm just going to impress you with this huge dot plot just to show you sort of the power of the single cell data set. So for those of you who are not familiar with the dot plot, what you're looking at is all of the subtypes or clusters on the left side. So these are all the different neuron types. These are all the transcription factors along the bottom that are expressed in neurons. And then the dots tell you, the size of the dot tells you the percentage of cells in the cluster where we detected the gene. So the biggest dot means that we found it in 75% of the cells. And then the color indicates the strength of expression. So what I want you to take away is that there's like some transcription factors which must be just common to making a neuron generally, but then you have these obvious groups of transcription factors expressed in different subtypes. And so that gives us a handle on trying to understand how these subtypes are made. So that's really the end of what I have to say about the nervous system. This is a pretty short section, so I don't know if there's like 1 or 2 questions before I move on. The last part is going to be about regeneration. But if there's any other things people want to ask about the nervous system, we could ask now.
We already have 2 questions.
Okay,
Matt McCoy is asking, I think you mentioned that cells migrate towards the head and foot, and that's where they shed. Does that mean those head and foot neurons are older than neurons in the midsection?
Yeah, so it's such a good question. So first of all, the word migrate's not quite right because they really are just pushed. So we call it sort of passive movement towards the extremities. And we know that neurons in particular areas have very particular gene expression. And so it's an excellent question. What does happen when a neuron gets pushed into the wrong area? And that's something that we really want to look at. So what we discovered during our study is that the epithelial cells are really phagocytic and that they actually eat up a lot of neurons. We found a lot of neurons, like in our supposed single cell data set, we found a lot of epithelial cells that had also neuron signature, and we realized, I won't go into the details, that the epithelial cells were actually eating up the neurons. So my hypothesis is that when the neurons get out of phase, they are just removed out by the epithelial cells. But we have not tested that or proven that anyway. But so the short answer is that, yeah, they are getting pushed, and they're getting pushed from the body towards the ends. And we know that there's a lot of stem cell activity to put new neurons into those neural nets where at the aboral and oral end where it's really densely packed. But what happens to the body column neurons when they get moved into that area, or when they get shifted out of place, it's still under investigation. Sorry, I hope that was clear.
Amy Lang asks, have you studied cancer in Hydra? What is a mutational landscape and transcriptomic differences in Hydra cancers? Also, what does cancer mean to an organism of such a high regenerative ability? Do Hydra die of cancer?
Okay, so I mean, this is not really my area of expertise, but I will say that there's one study claiming to have found tumors in Hydra. I don't know, and I don't know what the outcome is. I don't think they die of the tumors. I've never actually observed them in our cultures. I think, you know, the animals is so proliferative, it's almost like a little tumor itself, and it doesn't seem to, it seems to be able to deal really well with any kind of problems that might arise. You know, I think it can just get rid of cells that are damaged or could cause a problem. And so, yeah, the short answer is I don't really think it gets cancer, at least not readily. We haven't really looked at sort of like genomic level if there's damage happening and things like that. That's really unexplored in Hydra. And I'd be happy to discuss like that more in detail with the question asker later.
That's all the questions for now, and we have 8 minutes remaining.
Okay. All right, so in the last part of the talk, I want to talk about Jack's regeneration work. And so he wants to understand how injury triggers development. And the paradigm we're using is to cut the Hydra in half. So this is not only going to help us understand how injury triggers development, but how you basically are starting an injury from the same point. And so how does this part become a foot and this part become a head if you're starting from the same point? So looking into the old literature, there's actually a lot of information about how these early steps of organizer formation are established. I'm just going to very briefly review it. So when the animal is cut in half, actually what happens is that both sides initially get an increase in head potential. So even though this one is going to make a foot, initially this is starting to become a head organizer. But then the information from the surrounding tissue is integrated into the situation. And so in the bottom half, which is going to make a head, you get an amplification of this head potential. But in the part that will make a foot, you get a repression of this head potential. And so by 12 hours, the two organizers are totally set up. So you have a foot organizer here, and then a head organizer here, and then you will get differentiation. So we were really interested in this first 12 hours, like how are these organizers actually set up? And what is the molecular basis of these early observations? And so Jack's approach was to do sort of an RNA-seq, attack-seq time course, and we chose 0, 3, 8, and 12-hour time points for reasons I won't get into, but they seem to be useful time points. And this is just to understand the transcriptional landscape. So RNA-seq is just the transcripts that are coming on. The attack-seq looks at the chromatin structure. For those who are familiar, basically it shows you what areas of the chromatin are open at any given time. So he wanted to compare what was happening in the head to what was happening in the foot. And so he extracted all of the transcripts that are changing in head regeneration, and then all of the transcripts that are changing in foot regeneration. And he plotted these against each other, the fold change against each other. And what he found is that in the first 3 hours, anything changing in the head is changing in the same way in the foot. So it's just an injury response at this point. It's completely the same on both sides. And remarkably, the attack-seq is the same way. So the chromatin itself is also exactly the same in the first 3 hours. Then by 8 hours, you start to see specific differences between the between what's going on in head and foot for both the RNA-seq and the ATAC-seq. So what we found looking through the data is that actually a lot of head-specific genes are upregulated in that first 3 hours generically. So they're upregulated in both the head and the foot. And what was really cool is that we found lots of Wnt signaling genes to be part of this generic injury response. And Wnt signaling, as you know from earlier in my talk, is necessary and sufficient for head formation, but we're finding it as a general injury response, even if you're about to make a foot. So just you can focus on Wnt 910C because this is one of our favorite genes. In this part of the talk, red is always head regeneration and blue is always foot regeneration. So you have Wnt 910C coming up in the first 3 hours in head and foot regeneration, then coming back down for foot regeneration and continuing to go up in head regeneration. So this is a lot of Wnt pathway genes are doing. So, okay, the Wnt signaling is coming on in foot regeneration, but is it doing anything? Is it active? So we have some evidence that it is active, and one is that this is looking at the ATAC-seq data, the transcription factor binding motif for TCF, which is the downstream transcription factor for Wnt signaling. These binding motifs are opening up in both head and foot regeneration for the first 3 hours, suggesting that there is some activity of Wnt signaling in the foot regeneration. This plateaus in foot regeneration, but continues to open up in head regeneration. So I got excited about this because this early activation of Wnt signaling explains this old literature that a regenerating foot in the first few hours after injury actually has head organizer capability. So it's because of this Wnt signaling, we think that that is the case. So then we want to know, how is Wnt actually activated by the injury? And so we turned to our ATAC-seq data and we looked for transcription factor binding motifs that are open specifically at 3 hours. So we have a lot of different motifs that are changing, but we'll focus here on the ones that are open at 3 hours. And lots of these are transcription factor binding motifs, which should be bound by transcription factors which are known to be part of the immediate early response to injury, which is conserved essentially in all animals. We focused on one particular binding motif, the CRE, which opens up very dramatically here at 3 hours and then closes back down. And we found that also several transcription factors are upregulated that could bind this motif. So these are BZIP transcription factors. And what we found is that if we looked into our ATAC-seq data in the regulatory region for the Wnt 910C gene, we found a BZIP binding motif that was in an area of open chromatin. So these peaks are showing you open chromatin. This is the gene, and this is the upstream regulatory region. So if we just focus in on this open chromatin, what we find is that, so this light color here is at 0 hours. We found at 3 hours, you get this opening of this area, and then at 8 hours, it closes back down. For head regeneration, here's 0 hours, it opens, and then it opens even more at 8 hours. So this is not yet proof that this binding motif is partly responsible for this activation of Wnt 910C. We're making reporter constructs now, but this is, I think, really nice evidence in support of that. And so our hypothesis is that the injury triggers these very highly conserved injury responsive BZIP transcription factors, which turn on directly Wnt signaling genes. So we have this evidence for Wnt 910C, but also for Wnt3. So finally, and the very last thing I want to tell you about is this cool experiment that Jack did. So he made a prediction that prolonging the injury in the absence of the head organizer would actually cause heads to form. So if we have an injury and it turns on Wnt signaling and we don't have a head organizer already there, we should get heads, was the prediction. So Jack, and this is all Jack, this is his crazy experiment that he designed. It's one of those things where a student tells you something and you're like, okay, go ahead, that sounds insane, but you know, why not? And then to my shock, it actually worked. So what he did was he put a fishing wire through the middle of the Hydra, and then he left it there. In some cases, he took it out right away, and then for other cases, he left it there for 12 hours. And then he did this in a case where he took the head off and a case where he didn't take the head off. So the idea was that there should be this ramp up of injury signal where the fishing wire is going through the middle of the Hydra. And that could perhaps make a head if there wasn't a head already there. So when he left the organizers on, the animal just healed the wound, everything was fine. But when he had removed the organizers, he got, these are the most sort of dramatic examples, he got sometimes full heads growing out of the injury site. And now at this point, the heads, the original heads have also regenerated. So now you have a 3-headed animal. So these heads coming out the side is where the fishing wire was stuck through. Sometimes it was full head, sometimes it was just tentacles coming out. And so this happened even when he took the fishing wire out right away. But if he left the fishing wire in for 12 hours to really like amplify and prolong that Wnt signaling, he could get an increased incident of heads or ectopic tentacles coming out of those spots. And so the conclusion really is that the injury-induced Wnt signaling is sufficient to induce the head formation in the absence of the head organizer. And what we're really excited about is that the injury response seems to trigger Wnt signaling directly. So you first get sort of this default to making a head, and then the tissue context tells the animal, no, don't make a head, make a foot if there's already a head in place. And so this is sort of our first tentative connection between the conserved injury response and the homeostatic development of the animal. It's BZIP transcription factors. And there's some nice work from Rob Harris, who's at Arizona now, just started his lab there, showing this connection between BZIP transcription factors and Wnt signaling in a regenerating wing disc of Drosophila. So it could be a conserved connection. Okay, so with that, I would just like to thank the members of my lab. Here's our latest lab photo. Jack did all that regeneration work and also is the transcription factor guru in the lab. Abby is our nervous system person. So a lot of the nervous system work was hers, as I mentioned. Stefan is like the leader of the single cell project, so he has his hand in all of that work. Jeff is our fantastic collaborator who worked on the differentiation trajectories. The Robinson Lab is our neurobiology collaborators. And then I didn't get to talk about the work of some of these other fantastic lab members. So hopefully another time. And that's it. So I'll take any last questions.